The human digestive tract is not simply a single uniform tube. It is a series of drastically different biological environments, shifting rapidly from the highly acidic, oxygen-rich small intestine down into the dense, oxygen-depleted colon. The primary metric for microbiome repair is the initiation of a sequential, multi-species metabolic chain reaction. The first physical barrier is surviving gastrointestinal transit. The stomach acid and bile salts present a hostile environment, triggering chemical dissolution of incoming biological matter. Advanced formulas utilize a specific evolutionary workaround to survive this physiological stress, the viable but non-culturable, or VBNC state. The VBNC state is a form of dormant metabolism. Rather than expending energy on immediate reproduction, the bacteria enter a defensive posture, prioritizing the formation of impenetrable cellular armor. This protective dormancy stands in contrast to standard active probiotic cultures, which frequently degrade and die off before reaching their target site because they lack this structural defense. Protected by this armor, the VBNC state bacteria successfully pass through the stomach environment and arrive intact at the threshold of the small intestine. Arrival at the small intestine provides the biological wake-up trigger. The shift in pH, combined with environmental moisture, signals the bacteria to drop defenses and reactivate their metabolic processes. Utilizing the VBNC state ensures that the foundational architects of the ecosystem arrive perfectly intact and biologically ready to work. With the transit complete, the reactivated lactobacillus, acting as phase 1 of the G1 formula, begins operating within the small intestine. In this stage, lactobacillus performs aggressive environmental modification. It does this to neutralize a biological threat to lower colon ecosystem development, residual oxygen drifting downward from the upper GI tract. As detailed in panel 2 of this diagram, lactobacillus functions as an active oxygen scrubber.
It consumes and neutralizes that residual O2. The critical outcome is the active creation of a strictly obligate anaerobic, or oxygen-free, environment further down the digestive tract. This environmental preparation is the prerequisite step that allows oxygen-sensitive bacterial strains to survive in the next phase. Moving into the newly oxygen-free colon environment, strict anaerobes like bifidobacteria can now safely emerge and thrive. This triggers a process called metabolic cross-feeding. In this biological sequence, the biochemical exhaust of one species becomes the primary fuel for the next. Returning to the diagram, we see this chain reaction begin in section B. The bifidobacteria consumes the green lactate molecules left behind by upper tract lactobacillus. It then metabolizes that lactate, synthesizing an entirely new molecule, acetate. This acetate drifts further down the colon, where existing native commensal bacteria are waiting. These native commensals execute the final conversion step. They consume the acetate and convert it into terminal short-chain fatty acids, specifically butyrate. Butyrate serves a critical clinical function. It's the primary energy source directly nourishing colonocytes, the cells making up the gut wall. This direct cellular nourishment actively tightens the junctions between the cells, repairing and reinforcing the physical mucosal barrier of the gut. Mucosal repair is not achieved by the initial probiotic strains touching the wall directly. It is mediated by the downstream butyrate generated through this deterministic bucket brigade. A true ecosystem must also protect the walls it builds. This requires shifting from barrier nourishment to active barrier defense.
The Prolactor G1 methodology builds a biological environment rather than depositing isolated bacteria into the gut. It executes a sequence of biological pillars, surviving initial transit via the VBNC state, scrubbing toxic oxygen, executing the cross-feeding bucket brigade to generate butyrate, and deploying structural decoys for barrier defense. Achieving lasting gut barrier integrity is ultimately a problem of ecological engineering. It is solved only by building a complete, synergistic biological network.